Let's talk on thyroid gland. The thyroid gland in the neck is the largest endocrine gland that you have that's not mixed like the pancreas. The gland consists of two lobes and an isthmus. Two lobes and an isthmus. It has an extensive supply of blood vessels because what it has is inferior thyroidal arteries and superior thyroidal arteries. Inferior thyroidal arteries and superior. So a very large blood supply because the hormone must travel in large quantities in order to work on virtually almost every body system. On every body system. If we look at it in a histological aspect, that within the thyroid gland, it has these follicles called thyroid follicles. Here's one follicle. A follicle is surrounded by simple cuboidal cells. And in the middle is colloid. Colloid is an interesting substance. Colloid, in actuality, is your stored thyroid hormone. And this gland is the gland that has cells that do not have to store the product inside. They store the product outside the cells, extracellular. You have about a 2 to 3 month supply of thyroid hormone. What does that mean? If the thyroid gland stopped making thyroid hormone, you have about 2 to 3 months of stored thyroid hormone you can pull from. Okay? These particular cells, again, these cuboid are called follicular cells because they surround the follicle. The cells in between are called parafollicular cells. And the parafollicular cells secrete a whole different hormone known as calcitonin. Calcitonin. Calcitonin is involved with calcium, blood calcium level. Okay, so here again is a picture of the extensive blood supply of the thyroid gland. Here's a picture, photomicrograph of follicular cells with the colloid we just discussed. And colloid, again, stored thyroid hormone, is made up of thyroglobulin. Thyroglobulin is a glycoprotein that's made by the follicular cells. And iodine is attached, and we'll get into that shortly. Iodine is attached. So colloid is stored thyroid hormone, and this is it, thyroglobulin with iodine. Okay. The thyroid hormone we speak of is one called T4 and T3. T4 is thyroxine. It is also called tetraiodothyronine, and T3 can be called triiodothyronine. These are the two hormones that you would say are thyroid hormones. The term comes from the fact of the T4, you have 4 iodine atoms bound, and here you have 3 iodine atoms bound, and we'll discuss that too as to how this is made. Okay, right here is thyroxine. T4. 1, 2, 3, 4. 4 iodines. Here is T3, 3 iodines. Okay, so that's the structure. I'm going to come here. Let me come over here. Right here, I'm going to come over some. I'm going to make this a little smaller so you can see it. Right here is thyroglobulin depicted. This is a glycoprotein. Right here would be a tyrosine amino acid hanging off. Tyrosine amino acid. Okay? This is tyrosine. Right here is a picture of tyrosine. Tyrosine, you may recall, is one of our amino acids, 20 amino acids. It has it has this catechol ring. Tyrosine, anything derived from tyrosine would be a catecholamine, like norepinephrine, epinephrine, dopamine. But in this case, it's working differently. This is tyrosine. Right here, here is this tyrosine hanging off here. This is tyrosine right there, that amino acid right there. And if one iodine is hooked, iodine atom, it is monoiodine. Okay, if two, diiodine, and here's T3 and T4. So this gives you kind of an idea. You can go back and study this in the biochemistry section. So what happens is this. Here's this long glycoprotein, and as it folds, remember proteins have a primary, secondary, tertiary, and in some cases quaternary structure. But when this folds and two of these come close together, an enzyme can come in and hook the two together. Now, if both of them come in with two iodides, then it will form T4. If one comes in and only had one hooked to one with two, it would form T3. So again, you can go back and study this, but this is just to show how this, and remember where this is. This is in the colloid. This is the colloid, and this is just showing that. But we're going to go further, and you may get a better idea. Okay? So I come here now. The major metabolic hormone, so other words, thyroid hormone's action, it assists in the maintenance of blood pressure by increasing the amount of adrenal sympathetic nerve receptors. It regulates tissue growth, development of skeletal muscle nervous system, reproductive. But to say it in another way, it actually involves almost every system in the body. It affects metabolic rate, carbohydrate and protein metabolism, nervous system, cardiovascular system. It promotes normal functioning of the heart, muscular system, skeletal system, GI system, gastrointestinal. It promotes normal GI motility, and the reproductive and the integumentary system, the skin. Promotes normal hydration and secretory activity of the skin. So there are many functions that thyroid hormone does, many functions. Now, we're looking at the synthesis of thyroid hormone. So as we get into it, you will see that thyroid stimulating hormone will come and sit on the receptors of a follicular cell. It will then cause thyroid hormone to be secreted from the follicular cell into the circulation. Once secreted into the circulation, because this is somewhat lipid soluble, it will bind to a carrier protein called thyroid binding globulin. Okay, and then it will go to tissue sites that have receptors for thyroid hormone. All right. Whenever some is secreted, some more needs to be made to keep the same storage amount. Whenever some is secreted, because it will go in and take some from the colloid area, when some is secreted, some must be reformed in storage to keep the same amount. So that is where this story starts. Synthesis of thyroid hormone to restore that amount, to synthesize new thyroid hormone. That's where we are now. What I will do is to go step by step in the synthesis of thyroid hormone, but what I want you to do is to be able to study it also on your own using the text. Okay? So iodide is traveling in the circulation. Iodide, an anion. Iodide is pulled from the circulation. Here would be a thyroid follicle, pulled from the circulation by active transport. So this active transport pulls it into the thyroid follicle, and it's about 30 times more concentrated for iodide in here than it is in the circulation because of this active transport process, this active transporter. Once iodide comes in, it is then transported here. Now, when it is transported through this cell's cytoplasm, right here, what will happen is an enzyme will cause iodide to change into iodine. So that means it's oxidation, removal of an electron. Removal of electron. So now I have iodine. So we've pulled it from the circulation as an anion, iodide, pulled it through by active transport, okay? It's trapped, can't go back the other way. And now we're going to go straight through, and as we get here, what will happen is the thyroid peroxidase enzyme will then assist in converting it to iodine by oxidizing it and removing the electron. Okay? Now, so that's that step. Now, up here, as I started here, now I'm going to start here. Now we're going to construct the thyroglobulin molecule. Obviously, it would be in the DNA genes to do this. Then messenger RNA would read it. Messenger RNA would make it on the messenger RNA. Since it is going to be secreted, then it's going to, and it's a protein, it's going to be made on the rough endoplasmic reticulum. Remember, proteins are made on the rough ER. Go back to ANP1. Because it's going to be secreted, it is going to pass through the Golgi. The Golgi will sort, package, secrete, and also add the carbohydrates to it. Remember, I told you it was a glycoprotein. Then what will happen, the secretory product, which now these vesicles, transport vesicles, contain thyroglobulin, are then going to go and by the process of exocytosis, be liberated out into this region here, which is the colloid. So now what do we have? We now have this. We now have this here, which is the thyroglobulin, and that thyroglobulin has a bunch of tyrosine molecules along its primary linear chain. Then what will happen is enzymes will come in and combine iodine to the thyroglobulin tyrosines. In some cases, one will be attached, monoiodide. In some cases, two, di. Okay? Then what will happen is, as I showed before, enzymatic action then will come in and fold that chain around. Let me go here. We'll fold that around in such a way that enzymes will combine these tyrosines with the iodine once it's folded into the secondary structure, and then enzymes will allow the binding of that. Will allow the binding. If two tyrosines come together, both having two, it's T4. If one has one and one has two, it's T3. Okay, so we go back to this particular scheme here. So now we have this, and this is in storage now. This is in storage. So we now have several large molecules of thyroglobulin with iodine associated. Now, when asked for secretion because of TSH, then these will come in under endocytosis. Lysosomes will split them off. They will come in vesicles. Here's the lysosomes, and then be secreted as T3 and T4. So we'll close right here and take it up next.